Welcome to the deep dive. Today we are staring down a future where our collective digital memory, it's threatening to just overwhelm the physical world.
It really is.
We're wrestling with this problem measured in zettabytes, which is a unit of data so massive it's billions of terabytes.
And that's the global data crunch right there. The information we generate every single day, from social media, the cloud, AI, machine learning, it's not just increasing.
It's exploding.
It's exploding exponentially. It's a critical tipping point. And the numbers themselves, they just illustrate this impossible trajectory we're on.
So what are we looking at, say, by 2040?
The volume of global digital information is it's rapidly marching toward an incomprehensible level. And if we even tried to store all of that data using our current silicon technology,
It's not possible.
it's not. The sheer amount of raw material you'd need, specifically the silicon wafers, it would just vastly exceed the world's accessible supply. We are, and I mean this quite literally, running out of resources, space, and time.
Okay, so let's unpack this crisis, because it's not just about the capacity, right? It's about the massive technical and environmental cost of just keeping things as they are.
Exactly.
We always focus on the performance of silicon chips, but what's the unseen cost? The nuisance of their production.
Well, the environmental impact is staggering.
Hmm.
It's a cost we rarely, if ever, factor into the price of a flash drive or a server rack.
Right.
Take a typical modern chip manufacturing factory, a fab. It consumes approximately 20 million gallons of ultra-pure water.
Every month.
Every single month. Just to produce maybe 2 million chips. That's enough water to supply a small city, and it's all being diverted just to manufacture electronics.
And I'm guessing it's not just water. The waste from that process has to be notoriously toxic.
Exactly. The process relies on huge amounts of harmful, non-biodegradable chemicals. We're talking about cyanide compounds, silicon tetrachloride, arsine gas.
Things you do not want in your backyard.
Not at all. And they are not easy to dispose of. And they contribute massively to environmental pollution and, resource limitation. The reality is our traditional storage media are not just space hogs and power guzzlers.
They're an ecological burden.
It's a huge, unsustainable ecological burden.
And compounding this storage crisis is this sort of parallel crisis in computational speed. For decades, we all relied on Moore's law.
We did. The prediction that the number of transistors on a chip would double every 2 years, it gave us these predictable exponential gains.
But that's grinding to a halt now.
Oh, it is. That collapse is evident because we're hitting these fundamental physics issues as we try to shrink the transistors.
Yeah.
We're now talking about processes shrinking down to the 5-nanometer node.
And at that tiny scale, heat and leakage just become these huge, insurmountable problems.
Right. If you imagine a transistor as a gate controlling electron flow, when that gate becomes minuscule, the electrons, well, they just start behaving in ways we can't control.
They start to misbehave.
They do. We ran into a phenomenon called quantum tunneling. Electrons literally start jumping the barrier rather than waiting for the gate to open. This generates unusable heat, it causes leakage, and ultimately it limits the maximum density and speed we can get with silicon.
So the search for an alternative is now urgent.
Absolutely. We need a new medium not just for compute, but for storage itself.
And that search takes us away from traditional semiconductors and into these, radical new realms. Protein computers, optical systems, quantum computing, and the focus of our deep dive today.
DNA.
DNA.
Deoxyribonucleic acid. It's nature's gold standard. It is the most successful and efficient storage medium ever invented.
It holds the genetic blueprints for every living thing on Earth.
And now we're trying to harness that microscopic power to store our entire digital civilization.
So our mission today is clear. We're going to analyze the feasibility of DNA as a marketable, data storage medium. We'll look at its history, we'll dissect the complex chemistry needed to translate bits into bases, and we'll weigh the incredible advantages against the real-world challenges.
And ultimately, we're answering the question for you.
Is DNA data storage hope, or is it just hype?
Yeah.
Let's start just the absolute scale of this. When you hear that DNA is the most efficient storage medium, I feel like that's an understatement. We need to talk about the wow factor of these raw numbers.
Like you.
The sheer density. How much information can we truly cram into one tiny gram of this molecule?
The capacity it's truly unprecedented. Theoretically, DNA can store up to 455 exabytes.
455 exabytes.
Per single gram.
Can you just help us visualize that scale? 1 exabyte is a billion gigabytes, so that single gram is holding this unimaginable amount of information.
It is. To put this into a relatable context, This capacity is about 6 orders of magnitude. That's literally a million times denser than the best conventional storage medium we currently use for deep archives,
Which is magnetic tape.
which is magnetic tape. So if you consider that 1 gram of synthetic DNA could hold hundreds of petabytes of data, maybe 200 million gigabytes.
Right.
Imagine taking a massive energy-guzzling data center full of humming hard drives, hundreds of thousands of square feet of tape, and shrinking all of that information down to the size of a sugar cube.
That contrast really highlights the dilemma perfectly. Data centers need huge amounts of space, active cooling, 247.
And DNA is microscopic and passive.
So beyond the density, it's the durability that truly sets DNA apart. This is what positions it as the ultimate archival medium.
Longevity is arguably the most compelling feature. I mean, think about what happens to traditional media. Hard disks, magnetic tapes, flash memory, they all degrade, and they degrade quickly.
They have very limited lifespans.
Very. They often fail or just become obsolete within 10 years, which forces these massive data migrations or refresh cycles every 3 to 7 years.
Yeah.
Flash memory specifically, it degrades based on how many times you write to it, not its age.
But DNA is different.
DNA is what we call ultra-persistent.
And we have biological proof of this. This isn't just a theory, we see it in nature.
We absolutely do. Scientists have successfully retrieved and sequenced DNA from ancient samples. The oldest DNA ever recovered, it came from the bone of a relative of the mammoth found in Siberia.
And it was still analyzable after how long?
2 million years.
A million.
And we've also recovered DNA from fossils over 560,000 years old. That suggests that while our silicon storage becomes obsolete and physically fails, DNA has this inherent capacity to survive for millennia, as long as it has the right environmental controls.
Okay, but let me challenge that for just a second. That mammoth DNA, it was preserved in these ideal, naturally frozen conditions, right? An ancient freezer, basically.
That's a great point.
Is it really fair to expect that same longevity from synthetic DNA that we're storing in, a standard temperature-controlled vault?
That's a crucial point, and it's exactly why our preservation section later on is so important. Nature's best conditions, cold, dry, and dark, are precisely what scientists are aiming to replicate in a controlled, chemically fortified environment.
So a molecule itself can last that long?
The molecule itself can last that long, unlike plastic or magnetic tape, which simply can't. And this inherent stability, it also positions DNA as a truly sustainable, a green data storage medium, because it's non-volatile.
And because it's non-volatile, the whole power consumption issue just disappears once the data is written.
Precisely. Once the DNA has been synthesized and stored, it requires zero continuous power. No need for active cooling, which is a massive bottleneck in modern data centers.
That must drastically reduce the total cost of ownership.
Massively. TCO is a key business metric. It defines the overall cost of an asset over its entire life, including energy and maintenance. By eliminating those continuous expenses, DNA storage becomes economically viable over the very, very long term.
And that sustainability benefit must extend to data replication, which is a huge cost barrier for any business trying to maintain backups and disaster recovery plans.
That's a vital economic advantage. DNA has this natural, inherent ability for rapid, massive replication. Using what are called in vitro methods, specifically something called polymerase chain reaction, or PCR, you could just exponentially copy DNA sequences very quickly.
Okay, let's define PCR simply for everyone listening, since it's so key to the process.
Right. Think of PCR as a molecular photocopier. It's a method used in labs to rapidly make millions to billions of copies of a specific DNA segment.
So for data storage, what does that mean in practical terms?
It means you can make 1,000 copies of a single original DNA archive in about an hour for maybe $1.
$1.
$1. Now compare that to conventional backups, where every single copy costs you 100% of the original storage cost. This ability to get massive redundancy at minimal expense, it just solves a critical high-cost dilemma.
Okay, so if the theoretical advantages are so incredible, the density, zero power, cheap replication, this must have been recognized a long time ago.
It was.
Let's look at the history.
The idea of storing non-biological data in a biological medium is surprisingly old. It dates all the way back to 1988 with the work of an artist and collaborator from MIT, Joe Davis.
And he created something called microVenus. What did that actually store?
So Davis encoded a small piece of synthetic DNA. It contained a coded visual icon, a stylized image representing the Earth, female anatomy, and the number 0. And then he inserted this non-biological information into a live E. coli cell.
Wow.
It was a very early, very bold demonstration that DNA could host information that was completely foreign to the organism's own instructions. It proved the concept. The medium could be repurposed.
A great proof of concept. But when did we move from abstract art to, actual secure data?
It came in 1999 with Cleland and his colleagues. They successfully stored encoded words in DNA segments in the form of microdots.
And the significance there was the security aspect.
Exactly. They
Yeah.
introduced security and encryption. They created a key based on known PCR primer sequences and the 4 bases. So to retrieve the data, you needed that specific key to run the PCR and amplify the DNA. Without the key, the message stayed locked.
And we've progressed a lot since then.
Oh, yes. Recently, the scale has just grown exponentially. Researchers have managed to store data sizes between, say, 659 and 739 kilobytes. And that includes text, images, and sounds.
Which confirms its versatility for different data types.
Right. It proved DNA isn't limited to text. It can store multimedia, which is essential for deep archival.
Moving from a few encrypted words in the 90s to hundreds of kilobytes today is huge, but it is still small scale. The next big challenge is understanding the complex workflow that bridges the digital world of bits and the biological world of bases, at an industrial level. Okay, so this is where we shift from the potential to the practice. Moving data from a digital file into a DNA molecule is a complex, multi-step process.
It is.
We have to essentially filter massive amounts of digital information down into its fundamental molecular components and then, rebuild the code of life.
Absolutely. We break this down into the 5 core steps of what's called the DNA channel. First is encoding the data.
Right.
Second is DNA synthesis, which is the actual writing.
Okay.
Third is storage and preservation. Fourth, retrieval. And finally, sequencing the reading and decoding the information back into a usable digital file.
Let's start with that language barrier. Encoding and decoding. Digital information uses binary, base 2, zeros, and ones. DNA uses a quaternary system, base 4, A, T, C, and G. How do we translate between those two? And what are the crucial biological rules we have to obey?
So that translation process is handled by a sophisticated piece of software called a codec. It converts the bit stream into base sequences.
Okay.
And since DNA has 4 nucleotides, adenine, thymine, cytosine, and guanine, each one can theoretically represent 2 bits of information. So, 0, 0 might map to A, 1 to T, 10 to C, and 11 to G.
That's incredibly efficient. A single byte, which is 8 bits, can be represented by just 4 DNA bases?
Exactly. It's an elegant mathematical solution. But the chemistry, as you said, imposes these strict rules on the resulting sequence. You can't just randomly string bases together.
And that's the critical challenge.
It is. The encoding process is complex because the resulting DNA sequences have to adhere to strict biological and technical constraints. If we don't follow these rules, the quality of our storage just drops dramatically.
So what are the primary rules that the codec has to enforce?
There are 2 major technical pitfalls to avoid. First, sequences with what we call homopolymers.
Long repeats of the same base.
Exactly.
Yeah.
Long repeats, like poly G or poly C, they're catastrophic. During synthesis and sequencing, these long repeats cause the molecular machinery to sort of stutter.
And that garbles the data.
It garbles the data. It makes it really difficult to determine the exact length of the sequence. Second, the codec has to avoid sequences with very high or very low GC content.
So guanine and cytosine content.
Right. More than 75% guanine and cytosine is problematic because those bases bond more strongly than A and T, and that leads to technical difficulties in the sequencing process itself.
So the codec has to be clever enough to maximize information density while also ensuring the molecule is stable and compatible with the machines.
Exactly. And researchers are exploring ways to push that capacity envelope even further, beyond the standard 4-base alphabet.
How so?
Some are experimenting with mathematical tricks, defining what they call composite bases. For instance, they might define a new character M as 50 per CG and 50 per CT.
And that stores more data per length.
It does, theoretically. But it requires much greater sequencing depth to read it reliably.
And then there are the really radical ideas involving entirely synthetic DNA that doesn't exist in nature.
Yes, that's the frontier research.
Yeah.
It involves unconventional bases, often called xenonucleic acids, or XNA.
XNA.
These are synthetic orthogonal nucleotide pairs. The chemical structure of the backbone is modified, and researchers have synthesized several pairs designed to create a larger encoding alphabet.
With the ultimate goal being what? To store more than 2 bits per base.
Precisely. The aim is to increase the encoding alphabet size from 4 letters to 6 or 8 or even more. And that dramatically increases your density. But there's a major trade-off.
Of course.
Current commercially available sequencing technologies really struggle to read these unconventional bases effectively, which raises the cost and the error rate.
Okay, so once we have our perfectly designed, error-checked sequence, we move to the actual writing process, DNA synthesis.
We are physically building the data.
Right.
DNA synthesis results in short fragments called oligonucleotides, or oligos. These fragments are the fundamental units of DNA data storage.
And they're short, right? Around 300 nucleotides long.
Typically, yes. Because of the inherent limitations of chemical precision, if you go much beyond that length, the yield and the accuracy just decrease significantly, which means you need more error correction.
You mentioned there are two main competing methods for synthesis. What are they?
The traditional, well-established method is chemical synthesis. It uses phosphoramidite chemistry. This can be column-based, which is slow but highly accurate, or array-based, which allows for parallel synthesis of millions of different sequences at once.
And the newer, much hyped method
Is enzymatic synthesis. This uses biological machinery enzymes like terminal deoxynucleotide transferase to string the bases together.
And why is that considered the future?
It promises much greater speed, efficiency, and eventually lower costs than the chemical methods. Historically, the challenge has been the error rate. Early enzymatic efforts were just riddled with errors.
But that's improving.
It is. Technologies from companies like DNAscript have achieved a much more competitive error rate of 0.7% with their syntax technology.
Okay, a 0.7% error rate. That sounds tiny to me, but if a silicon chip had a 0.7% error rate, it would be useless.
Completely useless. And that difference is a massive bottleneck in adoption.
I can imagine.
Hard drives and flash memory operate with bit error rates measured in 10 to the minus 15. That's virtually zero error. A 0.7% error rate in DNA synthesis means almost every single oligo fragment you synthesize contains errors.
Which necessitates extreme redundancy and complex error correction codes.
Which we'll get to in a moment, yes.
Yeah.
But solving that synthesis cost and accuracy is critical because it currently represents the most expensive part of the entire DNA storage process.
So after the data is written into these synthetic fragments, we move to step 3, preservation and storage. How do you take something designed for a living cell and preserve it for millennia?
Well, as we noted earlier, synthetic DNA is highly vulnerable to environmental degradation. UV light, oxygen, water, heat, they all cause chemical reactions that result in breaks and mutations. So if we want thousands of years of stability, controlled, robust preservation is non-negotiable.
What's the gold standard for achieving that level of robustness?
The most effective strategy found by researchers is robust chemical conservation through encapsulation. This involves embedding the synthetic DNA in an inorganic matrix, things like silica nanobeads or magnetic nanoparticles.
Silica, like glass. What does that protective shell do?
Silica is inert. It provides an impermeable, microscopic shell that isolates the DNA from harmful elements like UV radiation and oxygen. The stability forecasts for these encapsulated solutions are just incredible.
How incredible.
DNA encapsulated in silica is estimated to last 20 to 90 years at just standard room temperature.
And if we use cold storage?
The stability just jumps dramatically. It's projected to last 2,000 years. It's stored at a moderately cool 9.4 degrees Celsius, and over 2 million years if you keep it at minus 18 degrees Celsius.
So we're essentially replicating and even surpassing the conditions that preserved that mammoth DNA.
Exactly.
Okay, let's move to step 4, retrieval and access. If this is for massive deep archives, access speed, the latency isn't primary, but you still have to be able to retrieve a specific file.
Absolutely. And currently, access is a challenge because it's inherently sequential. If you have a gram of DNA holding the equivalent of a data center, retrieving one file often means sequencing the entire pool of oligos.
Which is slow and costly.
Very. So to overcome this, the focus is on achieving random access, which is analogous to looking up a specific key in a conventional database.
How do you achieve that random access at a molecular level?
It's brilliant, actually. The indexing happens during the encoding phase. Researchers add specific addressable indices or unique PCR primers to each DNA fragment.
So the primer acts like a molecular barcode.
Exactly. When you want a specific file, you simply apply the corresponding primer key. That key only amplifies and selects the desired DNA sequences from the large pool, leaving the rest of the archive completely untouched.
So you only sequence the data you actually need. That's a huge time saver.
It is. This is why active research is focusing on using microarrays, digital microfluidic droplets, and DNA barcoded silica beads to improve that random access speed and efficiency.
Now let's go back to the elephant in the room, errors. Because synthesis and sequencing are error-prone, we need error correction codes, or ECCs, to ensure data integrity.
Absolutely. Errors, insertions, deletions, substitutions, they're inherent to the process. ECCs are paramount, and they're implemented by adding redundancy.
And that redundancy can be logical or physical?
Right. It can be logical, which is extra information encoded with the data, like a checksum, or it can be physical, just storing many copies of the same sequence. Logical redundancy is generally the goal because it uses less physical space.
So let's talk about some of these codes. How do they handle the specific errors we find in the molecular world?
Researchers use codes adapted from classic information theory, but they're tailored to the unique challenges of the DNA channel. A key example is Reed-Solomon codes.
Reed-Solomon.
They're applied in orthogonal directions to correct both single-point errors and what we call burst errors, where a whole section of data is compromised, maybe due to a strand break.
And that handles the integrity of the beta stream. What about just data loss? What if an entire oligo fragment gets lost during processing?
That's where fountain codes come in, particularly the Levy transform code. DNA storage is highly prone to deletion and loss of sequences during retrieval. Fountain codes are known as rateless codes because they deal robustly with this kind of error.
So you don't need all the original fragments to get the data back.
Exactly. You just need enough unique fragments. It's like having so many pieces of a puzzle that you can reconstruct the picture, even if some are missing.
And there's a critical trade-off here, isn't there, between accuracy and capacity?
This is the fundamental design constraint. The more redundancy you add, the more control bits and physical copies you store, the greater your error tolerance will be.
But you're reducing your actual information density.
You're sacrificing actual storage capacity for data integrity and durability. Finding that optimal balance, ensuring high accuracy without driving down the capacity too much, that is a central, ongoing challenge in DNA storage design.
So we've established that the fundamental science works, but moving from controlled lab experiments to a true industrial solution requires scale and, crucially, standards.
Right. This is where the biology world has to meet the business world.
And that convergence led to the formation of the DNA Storage Alliance in 2020.
A massive effort. It brought together 40 members, everyone from synthetic biology companies like Twist Bioscience to major tech giants like Western Digital. They later joined SNIA, the Storage Networking Industry Association.
What exactly is the alliance trying to accomplish? I mean, the technology is still moving so fast. How do you standardize without stifling innovation?
Their core mission is to create an open, interoperable data storage ecosystem. The goal is to define specifications for how different vendors' solutions interact.
So from the synthesis machine to the sequencing device.
To the preservation system, exactly. They're trying to create a standard digital wrapper around the biological heart of the process, ensuring, for example, that the DNA synthesized by Company A can be read by the sequencer from Company B.
So they're creating a shared operating manual for the entire pipeline. What are the first critical specifications they've developed to get this ecosystem started?
Two crucial standards have been introduced. The first is the Rosetta Stone specification.
The Rosetta Stone.
It's essentially the master boot record for a DNA archive. It's a mandatory preamble that defines how that specific archive was structured, what encoding schemes were used, what error correction was implemented.
And that ensures any sequencing machine in the distant future can read the data.
Even if the original company is long gone, it addresses format obsolescence beautifully.
Okay, so what about comparing the preservation methods themselves?
That leads to the second standard, the DNA stability evaluation method. This is a defined framework designed to provide an apples-to-apples comparison of containment systems.
So if one company claims that silica encapsulation lasts 2,000 years and another claims 5,000.
This method provides the standardized stress tests and metrics needed to verify those claims objectively.
Okay, let's pivot to commercial reality. Given the high latency and cost challenges we've talked about, what is the immediate, most practical use case for DNA storage right now? Where does it fit?
Well, it's not going to replace your hard drive anytime soon. The low latency and high cost mean it is currently optimized for deep archival.
That's the immediate, practical application.
It is. Statistics show that up to 80% of global data is cold. It's rarely, if ever, accessed after 90 days. DNA's density and longevity make it the definitive, sustainable solution for this massive, growing mountain of cold data.
So instead of wasting vast amounts of energy and space storing that cold data in these huge, power-hungry data centers, you transfer it to a few temperature-controlled freezers filled with these dense DNA capsules.
Exactly. And the high density and easy replication also make it the gold standard for disaster recovery. You cited the Universal Studios fire in 2008, where original artworks and masters were permanently gone.
Because the physical copies were lost or degraded?
Right. DNA offers a secure, gold standard backup. The fact that you can make 1,000 copies in an hour for $1 makes it cheap and fast to replicate and distribute copies geographically, preventing that kind of catastrophic loss.
And we are seeing commercial steps being taken, like Twist Bioscience spinning off Atlas Data Storage to focus on this deep archival tech. But the biggest innovation challenge is still the time and cost for that de novo synthesis building DNA from scratch. Has anyone found a way around that bottleneck?
Yes, and this is a revolutionary breakthrough that could drastically reduce costs. It's research from Arizona State University using what they call epibits, inspired by natural epigenetic modification.
Epibits. Okay, that sounds fascinating. Epigenetics is about how genes are turned on and off without changing the underlying DNA sequence. How does that translate to storing digital data?
So in biology, epigenetic modification involves adding or removing chemical markers, usually methyl groups, onto existing DNA bases to regulate gene expression. The ASU team adapted this natural process to encode digital information post-synthesis.
So wait, instead of slowly building a brand new custom strand of DNA to encode a file, which takes time and expensive chemistry, you're just modifying an existing universal template.
Precisely. Think of it as movable type. You have a fixed library of DNA strands, and instead of having to cast entirely new letters for every single book, you just chemically modify the strands after they've been synthesized once.
How does that create the binary code?
They use methylation to create a binary switch. A methylated base functions as an epibit 1, and an unmethylated base is a 0 way.
That fundamentally changes the economics of writing data.
It does. This method bypasses the slow and costly limitations of traditional de novo chemical synthesis entirely. It enables quicker, more economical solutions, and it significantly increases capacity efficiency by leveraging a fixed, pre-existing library of DNA that can be dynamically modified.
Which is critical for making DNA storage viable for faster archival tasks.
And for reducing the total time it takes to prepare a large archive. They were able to encode around 270,000 bits of data, achieving a rate of 350 bits per reaction. That's a massive leap in efficiency over traditional methods.
Even with all this rapid innovation in encoding and standardization, there are still significant challenges before DNA data storage becomes this ubiquitous industrialized solution. Let's focus on the two biggest hurdles: speed and cost.
Speed is the fundamental friction point. As we said, chemical reactions are inherently slower than electrical operations. Current DNA write and read speeds have high latency. We're talking minutes or hours.
So it's optimized for long-term archival storage.
Exactly. It just does not meet the nanosecond speed requirements for general-purpose computing. If you want to access a file in seconds, DNA storage is currently not the answer.
So how do we increase the throughput enough to make this technology appealing to major data centers?
The primary solution is enabling massive parallelism. We need systems capable of performing millions of writing and reading operations at the same time.
So it's not just about the chemistry, it's about the robotics and the fluidics.
Right. Technology providers have to increase the throughput of the underlying synthesis and sequencing, but also dramatically reduce the time it takes to physically move the molecules between steps. The actual manipulation of fluids in the lab is a huge time sink right now.
Okay, and what about the total cost of ownership, the TCO? The long-term storage cost is virtually zero because it's non-volatile, but that initial upfront investment has to come down to compete with magnetic tape.
And TCO is still dominated by those initial capital costs of synthesis and sequencing. Those steps remain the highest cost components. Advancement really relies on improving the core technology to achieve three goals at once: increasing the length of the oligos we can synthesize, improving the speed of the reactions, and maintaining or improving high accuracy.
And the current high cost of synthesis is the #1 thing limiting adoption.
It is the most important factor, yes.
And scaling all this up requires moving completely out of the human-staffed lab and into fully automated facilities.
Full automation is critical for large-scale industrialization. Most traditional DNA storage pipelines still require lab workers to manage fluids, transfer samples, run quality control.
But we're seeing progress there.
We are. Researchers like Takahashi and his colleagues have already demonstrated the first end-to-end automation of a DNA-based storage system. Achieving fully automated, high-density physical libraries that operate with minimal human involvement is really the final hurdle to commercial success.
What's truly fascinating to me is that the immense investment required to solve these problems is generating these powerful, unexpected wins that ripple out across other scientific fields.
It's an incredible positive feedback loop. The demand from the data storage world for highly accurate, cheap, and fast DNA synthesis is driving down the price of synthetic DNA universally, and that has huge ramifications far beyond data archiving.
So how does the push for cheaper, faster oligos for data specifically benefit, say, oncology research?
Well, take cancer research. Scientists use synthetic DNA sequences as custom primers or probes for diagnostic tools or for building targeted therapies. If the cost of synthesizing these custom sequences drops by half or more, which is what this research is promising, it drastically accelerates the pace and reduces the cost of personalized medicine.
So researchers can just afford to test more things faster.
Suddenly, they can afford to test hundreds of gene sequences and custom-built viral vectors in a fraction of the time and cost that was previously required.
And I'm guessing it's a similar story for synthetic biology and agriculture.
Exactly. Synthetic biology is the field of engineering biological systems. It often requires custom-built genomes or genetic circuits. If the building blocks, the DNA oligos, become cheap and fast to produce, it accelerates the development of everything from new biofuels to genetically enhanced, drought-resistant crops.
So the fusion of the semiconductor world and the synthetic biology world is creating these revolutionary, unintended technological consequences.
And they benefit life sciences globally.
So DNA starts as a passive storage medium, but the long-term vision, the ultimate frontier, is much grander. Where does this research ultimately lead, especially in terms of computation itself?
The ultimate frontier is DNA computing.
Okay.
This involves combining DNA storage with active molecular computing systems. So instead of DNA strands just sitting there passively holding data, they can actively participate in data processing.
So we're talking about using the molecules themselves as the processors, taking the silicon chip out of the equation entirely.
That is the long-term goal. DNA computing leverages the natural parallelism of chemical reactions. Specifically, it uses sophisticated mechanisms like DNA strand displacement.
Where one DNA strand kicks out another from a double helix.
Exactly. To implement logical operations, a molecular logic gate.
A molecular logic gate.
Precisely. By designing strands to interact in specific ways, you can implement general-purpose computations, molecular logic gates, even molecular neural networks. Instead of reading the DNA on a sequencing machine and feeding that data into a silicon chip, the DNA itself becomes an active participant in the processing.
That is a true convergence.
It is. Storage and compute residing in the same molecular medium, powered by biochemistry rather than electricity. It could eventually solve the computational speed crisis created by the failure of Moore's law.
We started this deep dive looking at the zettabyte dilemma, this crisis of exponential data growth hitting the physical limits of silicon and computational speed. And I think our analysis confirms that synthetic DNA data storage, it's not just a scientific curiosity, it is the definitive, viable solution for our long-term archiving needs.
It is. The unprecedented advantages, the extraordinary density, the non-volatility requiring no continuous power and durability measured in millennia, they ensure that DNA will augment and eventually succeed existing solutions like magnetic tape.
So it offers a practical and sustainable way to resolve that save versus discard dilemma.
Absolutely. The dilemma that every major organization faces when they're confronting these vast amounts of cold data, it is absolutely hope, not hype.
It's a compelling future to imagine, isn't it? Our critical digital legacy secured in these tiny, chemically protected microcapsules requiring zero energy and resistant to technological obsolescence.
And think about this. DNA molecules, the carriers of life's code, they are robust enough to potentially survive for millions of years, long outlasting any digital format we use today. So if we successfully encode our civilization's most critical scientific findings, our history, our art, and synthetic DNA today, what unforeseen, profound discoveries might scientists in the distant future, thousands of years from now, perhaps long after our current digital infrastructure has crumbled to dust on Earth as they sequence our preserved digital legacy?
Revealing not just raw data,
but a powerful window into the knowledge and the priorities we deemed most worth saving.